Hello, so come back to another video. Today we will solve Topical Answers 2023 paper 061042, which lasts for about 16 minutes and it has 80 marks and it carries 7 to 8 questions. Alright. So now let's look at this extended theory paper. Question number 1 says that red blood cells are specialized cells that transport oxygen, state substitutes, and red blood cells that combine with oxygen. So we all know that the red pigment or as hemoglobin, which combines with oxygen and forms oxyhemoglobin in order to carry out oxygen to all parts of the body. Right? This is the answer given written over here. Part B says state the name of the component of blood that promotes blood clotting. So the cells responsible for promoting blood clotting are platelets. Alright, there was a explaining that. Okay, it is clear as such. Now, on towards part C. Subsequently investigated the effect of immersed red blood cell with additional solution. They measured the diameters after the red blood cells and calculated the mean. Then they immersed each red blood cell sample in a different concentration of salt solution. After 2 minutes, they measured and calculated the two samples again. For 1.1 shows the result. So what did they do was they dipped the red blood cells in different salt solution of different concentration, and then they measured the change in the diameter. So the percentage concentration of salt solution is given over here, and the range is from 0.4 to 0.8. The initial diameter of the cells was kept at 7.5 micrometers. After a particular amount of time, the results were noted. In 0.4, the cells burst. In 0.8, the diameter increased, and in 0.9, the diameter did not change. And in 1.8%, the diameter decreased from 7.5 to 6.0. Alright? Part 1 says that the percentage increase in the mean diameter of the red blood cells that were immersed in 0.8% salt solution. The matches is still significant. So percentage change is always calculated by energy flashing. Final minus initial over initial times 100. Alright, so this is the working amount done for you at 0.8%, 7.5, 8.2. So because subtract 8.2 from 7.5 divided by 7.5 times 100, the answer will be 9.3%. Alright, now part 2 says explain the results for the first one immersed in 1.8% salt solution. So in 1.8% salt solution, the diameter of the cells decreased from 7.5 to 6. Why was this so? This means that the water has lost or water has moved from the cells to the outside of the cell to the salt solution, because the water potential in the salt solution was less than the water potential of the red blood cell, due to which the osmotic gradient occurred and the water moved out was cancelled the purpose of semi-terminal membrane. So I have read just within the answer value and make sure to use the terminologies around the water potential gradient and in the presence of semi-terminal membrane, because these are the IGCSE terms which will help you get those marks. Right. Coming towards part 3, state why there was no change in the mean diameter of the red blood cells of more than 0.9% salt solution. So whenever there is no change in the concentration or the size of the cell, this means that there is no net movement of water. This means that there was no osmosis taking place. Why was this so? This will only happen in the case if at only the concentration of the outside solution is equal to the concentration of the inside of the cell. This means that the solution is isotonic to the internal environment of the cell, which will result in no osmosis taking place. Alright, hope this makes sense. That is what I have written in this answer. The application of red blood cell is same as that of salt solution, but there is no net movement of water molecules. Alright? Now, part D says that state why red blood cells work and emerge in pure water, but plant cells do not. So we always know that plant cells have the cell wall outside or beside its cell membrane, which helps it to maintain its shape and maintain the turgor. Alright? So if kept in pure water, obviously the water potential of the water is very high. The water tends to move inside the cell to osmosis, right? So the cell membrane will burst in the normal cells, like in the animal cell, the cells will burst as compared to the plant cells, in which the plant cell wall will help them maintain that turgor. Alright? This is what I have written in this answer. Can you read it from there? Now part E says state to use of water in a plant. Because water has a lot of reasons on it. For example, it's helping in the transport of ions and sucrose from the source to stain and come to out parts of the plant body. Another use of the water is that it's solvents. Alright? So, it is also used in reacting in many of the processes like photosynthesis. Alright? This was the question number 1. Moving towards question number 2. So the question one is photograph some leaf of a water lily. There's a hydrophyte. Hydrophyte means the plant which loves to live in the water. The water lily has adaptive features that are on many different hydrophytes. But describe what is meant by an adaptive feature. So adaptive feature are a particular characteristic which is inherited, which helps the organism to survive. Alright, so this is a definition written, that inherited structural feature that helps the organism to survive or reproduce. This is a 3-mark question. 1 mark for telling that it is an inherited structural thing, 2nd mark for telling with that it helps the organism to survive in an environment. Right? Now, part B is the physical point of the question of the graph of the part of the leaf. So there are given a photograph having A part and B part, and then we have a C part. So we all know that A part is palisade nuclear layer because cells are very tightly packed and they're parallel to each other. Then part B has a lot of air space and they have a lot of cells that are regularly arranged. So this must be spongy nuclear cells. C is marking towards the long and large air spaces. Alright, so state the name of the parts labeled A, B, and C. So A is consisting neutrophil cells, B is consisting neutrophil cells, and C are the air spaces. Correct? So part 2 says explain how part C in 2.2 adapts and hydrophyte for its environment. Part C is air spaces. The air spaces are very large, right? So they reduce the density of the leaf, means they provide a resilience in the body of the leaf, which enables the leaf to float on the surface of the water and to get access to the large amount of sunlight over the top of the water for photosynthesis, right? So the usage for air space, I have written the answer over here to have this explained. Alright, so let's move towards part C. Scientists calculated the mean number of stomata per millimeter square in upper and lower epidermis in tomato plants and watermelon plants. Tomato plants are that of the district from. The 2.1 shows the results. A table 2.1 has plant, and they have measured the stomatas in the upper epidermis and the lower epidermis in both types of the plants. Alright. So we have tomato plant over here which has 10 stomatas and on the upper epidermis and 129 stomatas on the lower epidermis. Then we have water lily plants in which we have more 75 stomatas on the upper epidermis, and in the lower epidermis there are no stomatas. So we all know that the lower epidermis is facing the water, so there will be no need to prevent the water losses. That's why there are no stomatas or no guard cells present, right? Now, part 1 says compare and explain the difference between the mean number of stomata and a tomato plant and a water lily plant. First, have to compare and then we have to explain. So we can easily compare that. First of all, we will tell about the general comparison, general trend, that we can see that the number of stomata in the tomato plant is less than that of the water lily, that is 129 and 475. Then we can say and compare other parts of the epidermis, size of epidermis, that the tomato plant has more stomata than the lower epidermis, whereas water lily has more stomata than the upper epidermis. And we can say that water lily has 0 stomata in the lower epidermis as compared to the tomato, which has 139 stomata on the lower epidermis. So what I have written over here, then we'll explain the comparisons. Explain because why are the tomatas present in such a way. Tomato has more stomata on the lower surface. Why? Because it needs to reduce the water loss, right? It needs to reduce the water from beneath the surface of the leaves. That's why it has more stomata on the lower epidermis. Whereas water lily floats, there are no stomata in the lower epidermis because there is no need to prevent water loss. It is not to be considered, right? This is what I have written over here in the answer. You can read it from here. Let's move on to part 2. So the name of the cell that controls the opening and closing of the stomata. So we all know that guard cell is a part of the stomata which controls this opening and closing. Coming towards part 3, question 3 says that the 3.1 is the diagram for junction between the two neurons in a healthy person. The 3.2 is the diagram for junction between the same two neurons in a person who has Parkinson's disease. This disease affects the nervous system. So we can see that there are this is a one neuron is the second neuron, and the vesicles containing neurotransmitters are getting released from this neuron. The transmitter is then coming in the synapse, that is the junction between the two neurons, and then it is getting binded to the receptors present on the both sides of this neuron. Same way, we have the same neurons in the figure 3.2, but the vesicles containing neurotransmitters are very less, and the release of the neurotransmitters is very less, and the result, the binding of the transmitters to the receptors is also very less, right? They're asking us to identify the parts labeled X and Y. So X are the vesicle membranes, whereas Y are the junction between the two synapses that is known as between the two neurons that is known as synapse. We can see that they are marking towards this entire space. This is the synapse, correct? Parkinson's disease affects the neurons in the brain that are responsible for movement. Use information figure 3.1 to 3.2 and surgery to explain why the effect of the Parkinson's in a person moving. So we can see that in figure X and the figure 3.2 there are very less neurotransmitters being released. So the movement is going to be impaired and there will be slow reflexes. Alright. Why? Because fewer vesicular content neurotransmitters which are released across the synapse, and then there will be fewer neurotransmitters that will be binding to the receptor protein, right? And hence there will be fewer impulses generated in this neuron, which will slow or impair the responses. Correct? So this is the answer to this question. You can easily read it from here. Part C says strategy where the nervous cell control but different from the hormonal control. The nervous control is a very rapid process. It will just short level back. It involves neurons as compared to the hormonal. That is a much slower process, and it involves blood, and it provides long-lived effects. Alright. Now, part D says shape of the receptor protein is 3.1 of the important for the compounds when it comes to the receptor protein is determined. We all know that the receptor protein or any type of protein shape is determined because of based on the sequence of bases in their amino acid gene, in their gene. Alright, the sequence bases in their gene, and then afterwards sequence of amino acids and their structure will determine the shape of the receptor proteins. Right? This is the answer to this question of this video. Part 2 says cell membranes also contain the protein carrier to cellular protein carrier. The carrier proteins are responsible for the transport of the polar molecules like glucose, ions, and polar gases across cell membranes in or out of the cells through active transport or by using energy. So one mark for telling that they allow them to cross membrane, second mark for telling that they use active transport. Alright. Now, question 4 says the scientists investigated the effect of the temperature on the rate of photosynthesis in one species of the plant. But which involves enzyme control reactions. Discs were cut from and kept at different temperatures. The total surface area of the disc was kept same for each temperature. The volume of photosynthesis was produced, but the leaf disc was measured as used as the rate of photosynthesis. So what it did was they cut it. If this was a leaf, for example, this is a leaf, and they cut it the surface in the leaf and having the same surface area, and they kept them each for the same temperature, and so they kept the surface area same for each temperature. They sanitized surface area, right? Then they measured the volume of oxygen produced during the photosynthesis. And they plotted the results over here. You can see this in the graph that the temperature is given on x-axis and from 20 to 55, and the rate of photosynthesis of photosynthesis is given on y-axis. Alright, so we can see that what are the results? The results are from 25 till 35, the rate of the photosynthesis increased drastically, after which got to maximum at 40 degrees Celsius, it got to maximum at 50 degrees Celsius, we can see, and then it started to decrease till it decreased to 50 degrees Celsius, right? These were the results. Identify the optimum temperature. The optimum temperature is the temperature at which the enzyme shows their maximum activity. In this case, it is 40. Alright, so now part B says explain the results shown in figure 4.1. So the results, I just told you the results. Now we have to tell that why did we get such results. So we have to tell that enzyme control. This is the enzyme control reaction at 25 till before 40 degree. If we say so, the before 40 degree, the rate of reaction was less and was increasing because as the temperature is increasing, the kinetic energy of the molecules are increasing. So their frequency and receptor collision is increasing till 40 degree, at which the maximum activity of enzymes has been observed because there is the highest rate of photosynthesis and means highest enzyme substrate complexes were formed. After 40 degrees Celsius, what happened? The rate of reaction decreased because enzyme must have got denatured or have started to denature because of the increased temperature. The active site of the enzyme will no longer be remaining the complementary shape of the substrate. The rate of reaction is decreasing. Alright? This is how we will explain first before 40, then at 40, and then after 40. Alright? This was a 6-mark question. Part C says carbon dioxide was supplied in excess at each temperature during investigation. Explain why. So carbon dioxide was supplied in excess so that it may not get limited or it will not start affecting the rate of reaction. Alright, so to make sure that only temperature is affecting and not any other factor. We kept rest of the factors in excess. Alright. Now part D says suggests why not all of the oxygen produced by leaf is released. Because some of the oxygen is used in aerobic respiration and it remains in the air spaces. Alright. So let's move on to part E. Describe the role of chlorophyll in photosynthesis. So we all know that chlorophyll, that main segment, which is responsible for absorbing and capturing the light and converting it to chemical energy for synthesis of glucose in photosynthesis. Alright. Now coming towards question number 5, describe 2 ways in which circulatory system of a fish is different from circulatory system of a mammal. We all know that circulatory system of the fish is single circulation. It is transported or is removed from the heart only once in a single circuit, and it has no septum, and because there are only two chambers in the heart. Alright, so we are going to write down this in the answer. As compared to the mammals, in which the blood passes of the heart twice, one in the pulmonary circuit and the second in the systemic circuit. Now they're saying that explain the advantages of the double circulatory system. The double circulatory system prevents the mixing of oxygenated and deoxygenated blood because blood flows at high pressure to the body, and it enables a fast and efficient supply of blood to the rest of the parts of the body. Alright. Now, part C says figure 5.1 shows the parts of circulatory system of a mammal. So this is the part of the circulatory system, and the letters have been denoting the different systems of the body. And they are saying that state the letter of the 5.1 that identifies where the processes occur. So absorption of the products of digestion into the blood occurs in the gut. So this is part C. This is C, which is representing intestines, right? Excretion of carbon dioxide from the body is the job for the lungs. So we all know that lungs are present over here in the A. Now, then the next part is formation of a urine is done by obviously renal system, kidney, and the renal vein, or D is responsible for that. Production of bile is done by liver. So the liver system is B. Alright, now identify the name of the blood vessel labeled X. The blood vessel labeled X is this, that is taking blood from the gut or from the intestines to the liver. So this is always known as cardiac portal. Describe the role of the liver in excretion. So liver is responsible for detoxifying the toxic metabolites, alright, into the non-toxic one. So first of all, it has the job of the deamination, in which it removes the nitrogen containing part of the amino acids, and this removes excess amino acids from the blood. This regulates the concentration of amino acids running in the blood. So this results in the formation of the urea, and then urea is diffused out into the blood and travels to the kidney from where it is excreted. So this is the role of the liver in excretion. Alright. Now, question number 6 says state the name of the hormones released by ovaries. So we all know that estrogen and progesterone are released by the ovaries, and Part B says during pregnancy, antibodies are required by the fetus and the mother. State the organ the antibodies first to reach the fetus. The placenta is the only connection between the mother and the fetus in the womb. Alright. Part C says a baby was breastfed for 6 months. The concentration of antibodies in the baby's blood obtained from breast milk and the concentration of antibodies made by the baby itself was measured. But the results were recorded in the graph. As we can see that till 6 months the breastfeeding was stopped and the concentration of antibodies in the baby's blood was decreasing. And as it was decreasing in the blood, it was increasing, alright? So it increased maximum till 12 months. So they're asking that complete sentences to describe the changes. So we have to use the graph to fill in the blanks that are here, alright? We are going to do this, that after birth, the concentration of antibody required from the mother decreases rapidly from 0 till 6 months, alright? Antibodies start being produced by cells called lymphocytes in the baby immediately after birth, because lymphocytes are the cell responsible for the production of antibodies. Alright. Now the total concentration of antibody in the baby from both sources is 56 arbitrary unit at 4 months. Because at 4 months you can see that the concentration at this dotted line and the concentration at the solid dotted line. We have to measure the concentration from the graph and then we have to add it. Alright. Then we can see that the concentration of antibodies acquired from the mother and the concentration of antibodies produced by the baby were same at 3 months. Same means we have to record that value in this. Both of the graph lines are intersecting each other, which is at 3 months. Alright. Now, I hope this was clear. Now, part 2 is to describe the benefits of breastfeeding a baby for 6 months of life. So, the breastfeeding supplies breast milk is the best source of antibodies or the nutrients for the baby. Alright, so it is a good supply of the antibodies till baby starts making its own antibodies, and it provides passive immunity. Because passive immunity, why? Because it is giving directly antibodies from the mother to the baby, and it is preventing the baby from different sort of infections or pathogens. Alright. Now, part 3 says state 2 within which the breastfeeding, other than breastfeeding, that the baby can acquire immunity. So immunity could be acquired by direct injections of the antigens, which are known as vaccinations. Alright, that is active immunity, artificial, and then either we can also inject antibodies, that is artificial passive immunity. Alright, I hope this was clear. Now, part B says that explain the importance of the shape of an antibody. The answer to this question will be, we all know that antibodies are proteins, right? And they have different structures. They have different structures of the proteins, and this is according to the shape of the pathogen or the antigen they have to bind, which is complementary. Alright, so they are very specific in nature. So what is the importance of the shape? The importance will be that only particular antigen which has the shape complementary to the shape of the antibody will be binding to that antibody and will be getting destroyed. Alright, so this is the importance of this shape, that different shape. This makes the antibody more specific. Alright. Now, antibodies are proteins state the chemical elements present in all proteins that are also found in other carbohydrates and fats. So we all know that every biochemical molecule always have carbon, hydrogen, and oxygen in them, and rest, for example, nitrogen or sulfur is making them specific. Alright, so C, H, and O are present in all of the biomolecules. This was our paper. I hope this was clear, and if you have any questions, you can always comment in the comment section below or email me at the email address provided over here. And till the next video, goodbye and good luck.